So, this is one of the sort of delightful high points of the symposium. I'm going to just give you a little bit of an overview about the Boland medal and then introduce this year's recipient. So the Boland medal for the promotion of radiological protection. Boland, as I'm sure everyone in this room ought to know, was a giant of radiological protection and sadly died, but at a great age, 3 years ago. So ICRP wanted to do something to actually honour the memory of Boland. We decided in 2017, after discussion with, with his family, to establish this medal. It's to honour the memory and the dedication of Boland, who was a tireless worker for ICRP for many years. As you can see, he was the scientific secretary, a main commission member, he was the chair for 8 years, and subsequently up to the time of his death, an emeritus member too. Bo, of course, made many important contributions to radiological protection and the system during his career. He was one of the key players who had a central role in the development of the 1977 recommendations, publication 26, and it's really this that is the core of the foundation of our system of radiological protection today. ICRP decided to award the Boland medal to an early to mid-career individual. This is part of wanting to recognise the work of younger people, making a significant contribution to the promotion of radiological protection. One of the criteria was, in fact, that the recipient did not actually have to be an ICRP member to be considered for the award. So the medal is normally going to be conferred once every 2 years at our biennial symposium. And that's why, obviously, we're doing it here today. In fact, though, just because of the timing of Bo's death and the establishment of the medal, the first medal was awarded last year, 2018, on ICRP's 90th anniversary year. And it was nice to be able to present this in Stockholm because, of course, that was also the home of Boland. Just a little bit about the medal before you actually get to see one. The medal features Boland on one side. It has a map of the Earth and the world on the other. This is to represent the global reach of radiological protection that Boland achieved through his work. And the awardee's name is engraved below the bust of Boland. A little technical detail is, in fact, that it's a circular medallion. It is made of pure silver, and it is actually covered in or coated with 24-karat gold plate, and it is minted in Canada. You will see it, I promise, very shortly. But now I'd like to introduce the Boland laureate for 2019. This is Dr Liz Amesbury from the UK. And I like to do a little bit of background research and also a little bit of what I call nosy Parker digging to actually find out about the recipient of the medal. So Liz is a physicist by scientific training. She gained a degree in physics, Bachelor of Science, from the University of Bristol in 2003 and her PhD from Bristol University 4 years later. After that, Liz joined Public Health England, or as it was called, the Health Protection Agency. I can see she's smiling at the pictures that actually she didn't send me. I dug these out of an archive. And in fact, it seemed that very early in her career that Liz was sort of promoting the role of women in science. This is a local UK newspaper, the Northwich Guardian, which said, leading scientist does it for the girls, in fact. So it's good to see that not only Liz is promoting science, but actually she's promoting women within the field. Liz's current role is she is the Principal Radiation Protection Scientist and Cytogenetics Group Leader at Public Health England's Centre for Radiation, Chemical and Environmental Hazards. And she runs PHE's UK Chromosome Dosimetry Service. She is involved in several international projects, the UK NIHR Newcastle University Health Protection Research Unit Medical Radiation Theme, and has done much work with the EU concert-funded LD Lens RAD on the lens of the eye. And you're going to hear about her work in her lecture. Liz has many research interests: cytogenetic biodosimetry, the development of biological and physical dosimetry techniques and guidance, mechanisms and radiological protection aspects of radiation cataract, genesis and whole body acute radiation syndromes, and she also works on the academic aspects of leadership. She sits on review boards, governance groups at PHE, she supervises PhD students and lectures at several UK universities. She partakes in national and international committees, particularly Eurodos and NCRP, and has contributed to over 90 peer-reviewed publications. Liz likes to enjoy social life too, so she'll fit in well with the group here in Adelaide. Liz is a member of various organisations, research societies and institutes. She has already received awards during her career. She was the Melody Young Researcher Award recipient in 2014, and almost at the start of her career, the Institute of Physics Very Early Career Woman Physicist of the Year Award was given to her as well. This is the Nosy Parker bit coming up now. So, Liz is married to Martin. He's a financial analyst. She has 2 children called Meredith and Frederick. And what else is she interested in? Well, she likes running, very proudly has completed the London Marathon this year. She likes camping, and she is into vegan cookery. So it is with very great pleasure that ICRP awards the Lyndale Medal for 2019 to Dr Liz Amesbury. This is what the medal actually looks like, and I'd like to invite Liz to come to the stage to receive her medal. Hopefully see the medal. Liz, many congratulations.
Thank you very much.
Right, perhaps I now better... So, after all that, you're perhaps now going to hear a little of why Liz has actually been awarded this medal. So I'd like to invite Liz to give her lecture. It's called the Interdisciplinary Radiation Protection Research in Support of Medical Uses of Ionising Radiation. Liz.
Wow. Well, thank you so much, Claire. Thank you so much to the ICRP, to the organisers, the sponsors. I'm absolutely delighted to be here. I'm probably bright red right now, slightly embarrassed, still shocked but delighted to be this year's medal recipient, so thank you so much. I'm afraid I can't juggle or anything else or play the didgeridoo or anything, so instead I'm going to tell you a little bit about my research, and I hope I can live up to all of those wonderful things you were saying about whoever that person is, honestly. Right, so I have been in my career in radiation protection about 12 years now, and I've been privileged to work on a number of interdisciplinary research projects. So that's what I want to focus on today, and I'm going to tell you a little bit about some of the different projects. So apologies if there's a bit of jumping around, but I figured you don't often get to stand up here and tell people anything you want to, so you're going to have to sit there and listen to me. Thank you. This one? Aha. Okay, so as Claire mentioned, I run the UK's Chromosome dosimetry service, and so I'll start the talk by saying a little bit about dosimetry. And I stole this slide shamelessly from a colleague, David Lloyd, who managed the group when I started at Public Health England, Health Protection Agency as was, which lists some of the characteristics, not exclusively, of what we might be looking for in an ideal ionising radiation dosimeter. So, of course, we want something that's specific to radiation with low background, low donor variability, etc. We want it to be easy to do so we can train people. We want it to be cheap, ideally. And what we'd actually really like, is for it to be a risk meter as well. So we don't only want to know about radiation exposure, but what that radiation exposure means in the longer term. So we have a number of dose assessment approaches available to us, of course. Traditionally, we have dosimetry badges that radiation workers would wear. We also have what are known as fortuitous dosimeters, so these are materials that individuals might be carrying with them, which display some radiation response given the right circumstances. When individuals have large doses, as we'll hear a bit more about during this week, there are clinical signs and symptoms that we can use, so vomiting and diarrhea, etc. But most individuals who are exposed to ionising radiation are not exposed to high enough doses. And indeed, individuals can be exposed to ionising radiation and not be wearing their badges, or the badges can be exposed to ionising radiations and the individuals not. So we focus then on a biological indication of radiation dose. And for that, we use cytogenetic markers, so chromosomal aberrations and other changes in blood and other materials. So this is biological dosimetry. And of course, we do not yet have any single one perfect, ideal biological dosimeter, nor do we have a panel of dosimeters that give us all of the characteristics, but we have a pretty good approximation. So this is, again, a non-exclusive, non-complete list of some of the biological and physical retrospective dosimetry techniques available to us, and some of the different characteristics of them. So they have different responses in terms of time, I don't know how well you can read this, but specificity and sensitivity to ionising radiation. And they're at different stages of development as well. So most of my work, is focused on what we can do. I'm not very good at this thing at all. What we can do with the different biodosimetry methods. So one of the projects I've been working on over the last 5 years is in collaboration with Newcastle University, and HPRU stands for Health Protection Research Unit. It's a rather grand name, and I've been privileged to be the co-PI on the radiation theme of that project. And this is just a few words about the background. The idea behind this is that while we think we understand relatively well the sort of context for radiation protection in medical circumstances, there has been quite a lot of new technological development in the last few years. And certainly members of the profession, but also the public, are sometimes a little concerned as to whether we fully understand the longer-term adverse health effects. So what we were looking at were patient studies to inform this. How does radiation affect health outcomes? And what might we do to improve this? So we all recognise that ionising radiation in the medical context is hugely beneficial, but perhaps we can improve the way that we're protecting people at the same time. So just to put this in a little bit of context with the UK data, medical radiation, CT in particular, is the highest source of ionising radiation to the average UK individual. In the radiotherapy type scenario, we know that you don't get radiotherapy very frequently, if you ever get it throughout your life. But if you do get it, you'll get a high dose to a very localised region, and we know fairly well what to expect in terms of the acute radiation side effects, on a population basis, certainly. For diagnostic radiation, in contrast, these are much lower doses, people have them all the time. We know there are potential longer-term side effects, but they're much, much less well understood at lower doses. But it is also important to recognise that accidents do happen. So some of you will be familiar with the Mad River incident at a hospital in the US. A toddler came in with suspected head and neck injuries, and the CT technologist was not very well trained, so this toddler wouldn't sit still, so the technologist just kept retaking the scan until he was satisfied with the image. So this poor toddler ended up with 151 CT scans in 68 minutes, and it was a pretty horrible situation. So just because we have ionising radiation protection practices and guidance in place, it doesn't mean, of course, that these things are always followed. So the long-term objectives of the project, the radiation theme of the Chemical and Radiation Threats and Hazards project, were to better understand risks in the exposed populations in the context of other health risks. And of course that's important. Ionising radiation is not the only thing that causes health risks. Determine variability in clinical responses. So we're looking at individual responses here. And also, I've mentioned biomarkers, we're looking at identification of novel biomarkers of exposure if we can get our hands on some of those. And the longer term objectives, of course, were around providing more of an evidence base for safe use of low-dose medical radiation, particularly focusing on that low-dose region, and seeing what we can do with a more multidisciplinary approach, as has already been mentioned this morning. So I'm not an author on this paper, but it was done under the auspices of this project, and I really like to highlight the work that was done. This was a study of epidemiological analysis of cancer risks following cardiac catheterization. Quite a large cohort of patients under 22 years old, so these are children and young adults. And when this work was done, this is in the UK database of these individuals, the cancer rates were a little bit higher than expected. However, in looking into this, the epidemiologists that we work with discovered that a lot of these individuals happened to also have had transplants. And transplants were something that weren't routinely taken into account in the epidemiological analysis. When they did that, censoring for transplants, the standardized incidence ratio, the SIR, reduced to something much more along the lines of what was expected. This is a really nice demonstration of the fact that just sort of taking a step back and thinking about the data and thinking about what you're doing is able to sort of give us a nice answer there. I also want to highlight some of the work we've done on contrast agent. So there are a number of publications in the literature saying things like, well, if iodinated contrast agent is used, surely this will increase the DNA damage in the blood, and surely this will increase risk of cancer. Well, it's a bit of a leap, but probably worth looking at. So we looked at setting up some Monte Carlo models of the microscopic patterns of dose deposition within the blood vessels, and found that actually when you use iodinated contrast agent, the dose enhancement is only to a very small distance within the blood vessels. So contrast agents, yes, do indeed increase dose to the blood and the vessel wall, but do not increase dose, or are very unlikely to increase dose to the surrounding tissues. So again, it's a relatively small piece of work, but targeted, looking at a specific problem, something that had been highlighted in the literature. What can we do to try and answer that question? And then another example of using the data from the CT scan cohort that was already in existence. I'll say a little bit more about cataracts later, but the idea was to evaluate the risk of cataract in these individuals. So these are the young adults that I mentioned already. So colleagues at the University of Newcastle looked at the number of scans that individuals had had. They looked at the type of scan. They looked at how the scan was set up, all of the associated data, and identified that although the majority of individuals have a relatively small number of scans and therefore a small dose, when the lens was included, there were individuals who potentially received lens doses above this threshold, assumed threshold of 0.5 gray, and indeed one individual got a cumulative lens dose of 4.7 gray. So, of course, the CT scan was carried out for something totally unrelated to the lens, but it highlights the need to consider lens dose in all circumstances. Okay, so I'm going to jump again and talk a little bit more about biomarkers. And I really like this slide. This comes from the Perno publication of 2012, looking at biomarkers for molecular epidemiology in radiation research, and it has been updated. But there are so many different things that we understand that ionising radiation does on a biological, biochemical and physical level. And so many candidate biomarkers out there that we're only just beginning really to try and exploit these. So again, under the auspices of the HPRU project, we've been looking at developing the existing and new biomarkers specifically for relevance to these medical radiation exposures. And we've looked at a large number of candidate biomarkers. We, in parallel with the project, had some funding from the NIH, the US, Centers for Medical Countermeasures Research, and we had a project which we called RT-Gene, to try and look at whether coding or non-coding RNAs might be able to be used to form a radiation-specific biomarker, either in combination with some of the traditional cytogenetic markers or alone. So we have blood samples from approximately 20 large field radiotherapy patients. And in terms of the cytogenetic data, we did see validation of the cytogenetic dosimetry, so the well-established emergency response spider-symmetry techniques that we have worked very well in this patient population. We also developed a new blood dosimetry method, and we developed some new statistical methods to calculate partial body doses, because of course these individuals don't receive full body doses. And the figure just demonstrates the nice relationship between our estimated and estimated doses using two of these different types of estimation methods. The most exciting data is in the gene expression work that I mentioned. And this is a very unclear slide. You probably can't read any of that. But we had samples at the start of radiotherapy, through the middle, and towards the final fraction. And although there's a big mess here in the large number of genes that we looked at, we do see statistically some genes upregulated and some genes downregulated. And that statistical significance only arises when you look at the panel of genes together. So that's interesting. This is in preparation for publication, so I'm not going to say any more about that just yet, but we have this data plus data from a number of other studies that we're putting together, hopefully to reach very soon a critical mass of data which tells us that we have a genetic biomarker of radiation response in patient groups. That's very exciting. Okay, so everybody loves a slide with large lists of publications on it. So we've done lots of publications, we've been to lots of conferences, we've talked to lots of medical stakeholders. But the thing that I really want to say about this is that we've had rather a large component of public and patient involvement in this work. Now, when I started with the Health Protection Research Unit, PPI, as we call it, was something that was relatively new to me, and I didn't fully understand the importance of it. But actually, I'm a total convert now. So the justification for me is that if we're doing publicly funded research, we should be able to ask the public if they agree with what we're doing, and they should say yes. And if they don't, well, we need to convince them or we need to do something else. So that's how I look at it. So we've had a number of different events focusing on different aspects of the project, and at one of these we actually gave them a number of presentations and gave them a little questionnaire at the end and said, do you agree with how we're spending money on the project? And they all said yes, so woohoo, that's good. Very happy about that. We're also working with members of our public engagement panel and some artists with the idea that we might do something a little bit different around artistic interpretation of the scientific research based on the public's interpretation of what we've done. This is a bit of a work in progress, but we're going to be looking to do that at an event that will take place, fingers crossed, towards the end of March. I have a teleconference next week, hopefully, to finalise the date, but everybody will be invited. That will be a stakeholder dissemination event for a number of different projects, so watch this space. And just to sort of round up the HPRU work, that project will come to a close within the next few months, but we already have a new pilot project, particularly focused on vascular surgery operators and patients, so looking specifically at longer-term risk of these, again, relatively low doses in the short term, but over a long period of time, the operators can build up relatively high doses. So looking specifically at modelling for aneurysm repair, we do see that patients, in this case, get high doses, but that's justified because the procedure saves lives. That's perhaps nothing particularly new in the conclusion, but we've got some nice data there, and a lot more coming on biomarker work and epidemiology follow-up in these individuals as well, going forward. Okay, I'm going to say just a few words about emergency response now, so we're shifting again to another different topic. Many of you will be familiar with the RENEB network, so this now stands for Running the European Network of Biological and Retrospective Physical Dosimetry. It was a funded project under Framework 7 from about 2012 to 2015, the idea being that we need to create a state of readiness in terms of consistent and accurate biodosimetry and physical dosimetry procedures in a network across Europe and beyond. And indeed, the RENEB Association was then founded following the close of the project in 2017, and we believe we are well prepared to do this. So if there's a large-scale accident or incident and biodosimetry is indicated, then we are ready to provide that dosimetry. In addition, many of you will be very familiar with the European Radiation Dosimetry Group. Working Group 10 is the group that focuses on retrospective dosimetry, and I've just been elected chair, so that's very exciting. Looking forward to taking that forward. The motivation for Eurodos Working Group 10 is to establish a network to do all of these things, to make sure we have validated existing and developed new biological and physical dosimetry methods, and think about how we can put these things together to, again, answer that question of the ideal dosimeter that I mentioned at the start. But what I really want to highlight about this is that although we have two networks that nominally are working on a very similar topic, they do have distinct aims. So RENEB is currently focused on the emergency response capability and capacity. EURODOS Working Group 10 is novel research development in comparison with and standardization in terms of the existing dosimetry methods. But we have a huge amount of overlap between these networks. So we have, in effect, a network of networks. So EURODOS and RENEB work together to do intercomparisons. These are crucial for validation of the methodologies, literature reviews, training, etc. So, it's another example of how working across the disciplinary boundaries and across the formal network boundaries can really lead to additional things. And this is one of the things that I'm very proud of within the groups. So I still am running the uncertainty task group within EURODOS Working Group 10, and I've been very active in RENEB as well. So we've had a couple of joint activities between RENEB and EURODOS. In 2017, we produced a review of the uncertainty estimation methods that are used across our standardized methods. And then, well, that's fine in itself. A nice review is great for everybody to read in the literature, but what we really wanted to do was develop that further and come up with best practice for uncertainty estimation within those biological and physical dosimetry. So we ran 2 short courses funded by Concert in collaboration with EURODOS, which focused on how to actually do these things. So we trained a number of scientists, early career scientists there. And indeed, development goes on, so there's now a shiny app in R that everybody who's interested in uncertainty in retrospective dosimetry is welcome to contribute to. A few of us are on the list already, but it's an open system for contributing. The idea being that we'll produce software tools which are web-based so that everybody can use these things and everybody can use that best practice that we've all agreed. And just to highlight, not only in the uncertainty estimation, but in the practical methodologies of using these biodosimetry techniques. So premature chromosome condensation is a cytogenetic method that allows us to relatively rapidly look for chromosome aberrations within blood, within the lymphocytes. It's been used within the field for a number of years, but within the last couple of years, one of the scientists in my group received some training from one of the RENEB experts, and we've now implemented this and are standardising it and refining it at Public Health England. So, we've been properly working together, which is really nice. Okay, I'm going to say just a few words now about radiation cataract to round things off. So I couldn't get away with giving this presentation without including at least one of the ICRP publications. So 118 is the publication of choice. And I'm sure I don't need to say to this audience that up until relatively recently, it was the accepted paradigm that radiation cataract was a deterministic late effect. So there was a threshold of approximately 2 Gray for acute exposures, below which it was not expected that ionising radiation would induce cataracts. However, there's been a huge amount of work actually in recent years, epidemiological analyses and reanalyses and all sorts of things that have suggested that the lens is more radiosensitive than previously thought. And this has, well, in terms of radiation protection, culminated in the new European Basic Safety Standards. So the threshold for radiation cataract is judged to be about 0.5 Gray, and correspondingly, the occupational lens dose limit was reduced to about 20 millisieverts per year. But, of course, there's always a but, this is almost solely based on epidemiological data, certainly at low doses. We don't understand mechanistically how ionising radiation might be influencing cataract development. So we have some key questions here, and in 2017, again it feels like a long time ago now, we set up the Lens RAD Project, which is an EU concert-funded project to try and answer these particular questions. So how might low dose rate, in particular radiation, be involved in development of cataract? what's dose and dose rate doing, but also how might genetic background and age and sex and some of the other factors be involved. And as we were looking at lenses, but we're going to be exposing animals and thinking about what this really means for radiation protection, we also wanted to try and answer the question of could the lens be used as a global biomarker of radiosensitivity. So it's a relatively ambitious project. This is a very complicated slide which has lots of information on it. It's a very ambitious project, as I said, but the idea is that we have exposed animals and cellular models to various doses and dose rates. We have some short and long-term endpoints, so in the sort of hypothesized chain of reaction of cataract formation, we're looking at various different things, and we put all of that data together with some statistical modelling to try and answer those research questions that I've already highlighted. I'm just going to show you a couple of the results. There's a huge amount of data. I'm delighted to say, coming out of this project, we are hoping to publish a special issue of a journal, yet to be finalised, but within the next year or so, with all the different outputs from this project. But just to highlight very briefly that, as expected, everybody knows this, right? The longer you live, the more likely you are to get a cataract. So we did see lens density increasing with age. When this particular model of mouse, which is deficient in DNA damage repair, was exposed at 10 weeks, we didn't see significant radiation-induced effects. But when we exposed them at 2 days, so these are neonates, we see significantly increased risks of cataracts as the mice age, associated with dose and dose rate, and if we put that with the wider data, also with sex and genetic background as well. So all of these things are working together to give the overall radiation risk. Also, to answer this sort of intriguing question of whether or not the lens might be a global indicator of radiation effects, we have data from the INSTA project, the Precursor project, together with the new data from the Lens RAD project that shows that yes, ionising radiation, dose rate, but also working together, interacting with gender and with sex, and with the other factors, does impact neuronal progenitor stem cell, various things there, and also the behaviour of the mice too. Okay, and then so bringing it back to the stakeholder involvement and dissemination. We did quite a lot of stakeholder mapping around this project. And, well, as you can expect, we have a large number of stakeholders that we identified. But what we tried to do within Lens RAD was think about who we could really reach with the outputs of the project. So we have a number of different facilities available to us. Some people read the basic safety standards, and everybody reads the ICRP reports, obviously. Then publications, but also we needed to reach people who are actually using ionising radiation. So the Royal College of Radiologists have been interested, for example, and members of the public too. So I mentioned at the start we have this event that we're planning, and we're planning to invite lots of people to hear about what we've been doing, and hopefully they will be interested. Okay, so that is the end, pretty much. Good timing. So I don't think I've written anything remotely controversial here. I think everybody in this room would agree with everything and say, well, it's fairly self-evident. I hope so. But the work that I've been privileged to be involved in has demonstrated that all of these things are true. I think that's what I'd like to say. So the work that we do, the research that we do in support of radiation protection needs to be driven by scientific need, of course, but also by public perception of that need. That's really important, that we're communicating what we're doing effectively enough to convince people that we're doing something worthwhile. All of the work that I've shown you has had a large amount of input from early career scientists and has been genuinely interdisciplinary. So radiation biologists and epidemiologists, but also physicists, clinicians, members of the public, all sorts of different people. And so putting all of that together and across various different projects will really allow us to draw those concrete, might be a strong word, but conclusions. So going forward, integrated interdisciplinary research will be hugely important, as will the stakeholder engagement. So we've gone from public and patient involvement to medical, public, patient involvement and engagement and dissemination, and you can probably add many more letters to that as well. And probably most of what I do will remain focused on this emergency response work in terms of biomarkers, but I hope I've demonstrated to you that there are a huge number of sort of applications and we can work across disciplinary boundaries to try and answer some of these important questions. So I've got 17 acknowledgement slides now, I'm afraid. I have not named everybody. I'm very sorry to anybody who isn't named, in particular the Eurodos and RENEB colleagues. I started to think about it and I thought, you know what, there are just too many of you. So thank you to everybody, and apologies, your names are not there. The HPRU project participants, and I already had the LD Lens RAD slide, so thank you so much to the project partners and also the advisory board members who've been really crucial in keeping us on track, focused on mechanistic research, but in support of radiation protection. And, you know, at the end of the day, it is all about the people, so I wanted to show you this lovely picture of my group at Public Health England with our Christmas jumpers and Christmas glasses on as well. If you want to hear more, you can go and look at our website, but also please do talk to me here, email me, be delighted to hear from you, and I better stop. Thank you so much again.